There was more gold than you can imagine found in a tunnel under the Vatican City. Running from Vatican City, and deep underground, there is a tunnel that runs almost in a direct line to Jerusalem. The actual direct flight distance is 1,432.90 miles, 2,306.03 kilometers. The length of the tunnel was given as approximately 1,500 miles, so it cuts through underneath both land and water. Starting from the Vatican City, for the first 150 miles, 241 kms, there was found a huge amount of gold stacked 13 levels high. I'm not sure what 13 levels high actually means, but whatever, it was a lot. Along with this massive amount of gold were found many books or manuscripts. It has been said that these ancient books were stolen out of the Bible. Do I believe that? Well, I'm not going there as it would be conjecture at best. I'm just reporting what was relayed to Charlie Ward as to the findings under the Vatican. You may remember that when the COVID-19 pandemic first hit, one of the worst places was in Italy. Remember all the headlines. COVID-19 pandemic in Italy. Italy scrambles to contain second wave as daily COVID-19. Italy's daily COVID-19 cases hit new high. And on, and on, on. This was when the alliance and military were taking down the 13 bloodline families, and the Vatican. During this period they found the underground tunnels, the gold and the scrolls/books etc. It was completely necessary that they kept the world's attention on the virus, which allowed them to clean out the tunnel and other necessary work that needed to be done. 650 aeroplanes were required to carry away all the gold that was brought out of the tunnel which led to Jerusalem. Please note that when they went to Buckingham Palace, they found similar, huge amounts of gold etc., stored up for the wealthy, having been robbed from the poor. It was all taken to Fort Knox for safekeeping. When Nizaragasara are being rolled out, much of this gold will be used on humanitarian projects. There is more than enough gold in the world to back all the world's currencies on a one-to-one basis, and sufficient to take care of all needs worldwide. We are moving into a different world, only made possible by the complete removal of the evil cabal who have been busy robbing us blind in every possible way. It raises so many questions, doesn't it? The love of money is the root of all evil, the Bible quotes in 1 Timothy 6:10. Please notice it's the love of money, not the money itself, that is the root of all evil. Loving money, and ever being greedy for more money, which brings with it power, which ultimately leads to total control of others. Does this give a full picture of what the New World Order have been working at over a very long time? So there was more gold than you can imagine found in the tunnel that connected the Vatican with Jerusalem. How did it get there? Who put it there? Over how many thousands of years has it continued to be stockpiled? Will we ever know? Who knows?
November 25th, 2019, Dr. He Jiankui announced via YouTube that he had decided to push humankind across a controversial threshold. This announcement was, of course, that his work had resulted in the birth of twins who had both been subject to genetic engineering using CRISPR. This was a threshold widely considered to be off limits. In 2015, two of the foremost journals in research, Science and Nature, both made similar statements stating that modification of human germ cells, that is sperm and eggs, should be avoided. The scientific community seemed to have come to an agreement not to experiment with this technology until we could agree how and when to apply it to humans. But Dr. He Jiankui has unceremoniously dragged us across this line in the sand, whether we like it or not. Now, more than ever, there is an urgent need to stop and ask what the outcomes of this work are for the individuals who are subject to it and how the rest of society may be affected. To understand all this, we're going to need to understand how CRISPR technology works and what the intrinsic shortcomings of this technology are. DNA is made of nitrogen and carbon-based rings, which form 4 distinct chemical codes: adenine, guanine, cytosine, and thymine. These codes form an easily reproducible double-stranded molecule, which is read in groups of 3, called codons, which are contained within structures called genes. These genes are simply the parts of the DNA that code for the more structurally complex proteins, which in turn form the biological machines and structures that are required for all life on Earth. Due to DNA's ability to copy itself, till now, the code in the DNA of all humans can be traced faithfully to the first cells on Earth. Dr. He's work changed all of this by employing the use of CRISPR and Cas9 technology to purposely change the DNA code in the 2 twins referred to as Lula and Nana. As its name suggests, the CRISPR-Cas9 mythology is dependent on 2 parts, CRISPR and Cas9. CRISPR is a kind of repeating DNA structure that was first discovered accidentally in bacterial DNA by Japanese scientist Yoshizumi Ishano in 1987. For 17 years following this discovery, the exact function of these DNA repeats went undiscovered until it was identified as the means by which bacteria identify and protect themselves against invading viral DNA. It turned out these strange DNA repeats actually came from viruses. Bacteria have the ability to cut up invading viral DNA, store it in their own DNA, and then subsequently use it as a guide system for the enzymes Cas9, which destroys the viral DNA sequence should it invade again in the future. In order to do this, Cas9 uses the stored DNA to make something called the guide RNA. This guide RNA is usually coded for by a CRISPR repeat and guides CRISPR to DNA it should cut. There are 2 conditions for CRISPR's binding to its target. First, Cas9 must bind to the target sequence and a specific 3-code variation in DNA, which is called a PAM. Most PAMs can be recognized by Cas9 as they contain the code GG. These recurring patterns are targeted by a bacteria's Cas9 system as they identify the DNA of invading viruses. This gives the bacteria an ability to target and destroy the viral DNA. However, in the last 5 years, we've learned to leverage this ability to direct Cas9 with guide RNAs of our own design to alter the genome of other organisms. The guide RNA binds to a part of Cas9 called the alpha helical lobe. In turn, another domain of Cas9, the nuclease lobe, binds to any flanking part of the DNA strands that matches with the guide RNA, causing the double helix structure of the DNA to unwind. This process is facilitated by the proximity between the DNA and the Cas9 complex and electrostatic interactions that occur between the RNA and DNA. At this point, 2 specific domains of the nuclease lobe cut the DNA. Although we're not fully sure how this mechanism occurs, comparative studies suggest that the most likely way is that the 2 separate parts of Cas9 are able to rip from water molecules, and in turn weaponizes the deprotonated water molecule to break the DNA apart. This is done at 2 sites in the DNA, one which cleaves the DNA at the target site, and another which cleaves the DNA at a non-target site. At this point, a number of things can happen depending on which approach is needed. Cas9 can be modified to include a deaminase enzyme which can target specific bases for mutation, leading to brand new mutations. A process known as homology-directed repair can be used to insert new DNA at the cutting site, or the targeted piece of DNA can be simply cut out and removed if it is deemed to be undesirable. Dr. He Jiankui opted for the later option and simply cut a section of a gene called CCR5. The section that was cut corresponded to a mutation known as Delta 32 that confers its owner with protection to certain strands of HIV. Dr. He's data seems to suggest he may have artificially induced a mutation, similar to but not identical to the Delta 32 mutation in these twins. In order to do this, Dr. He had to first optimize the gene editing protocol in mice, monkey, and human cells. The reason for this is to make sure the protocol has high specificity, simply put, that the gene editing technique was editing the target DNA and not some other sequence. This claim is, of course, key to the validity of the experiment. Unedited embryos can be thrown away after all, but for obvious reasons, making unspecific alterations to a human's genetic code would be a major red flag, and Dr. He's work raises major red flags for this exact reason. Their check for specificity of gene editing used parental genetics as a reference point, an inadequate approach due to the fact that a parent's genetic material is subject to a certain degree of randomization every time a sperm or egg is produced. It's further complicated by the fact that no two sperm or eggs will be the same, and that their morphology did not account for the possibility that larger sections of the genetic code could be deleted by their implementation of the CRISPR-Cas9 system. Further to this, from the data released so far, we can tell that the mutation induced in the twins does not exactly mirror the Delta 32 population that He is attempting to induce in the twins. Although the protein produced should, in theory, largely mirror the naturally occurring mutation, we cannot tell that for sure because it has not been tested. Standard practice at this point is to test these mutations in an animal model in order to assess unexpected effects of the experiment. This is perhaps the most troublesome part of this experiment. Lulu and Nana are test subjects for a genetic mutation that we have not even taken the time or care to test in animals. Nonetheless, the mutation was induced in a total of 31 human embryos. These embryos were grown for a period of 5 to 8 days, and the 19 that were deemed normal were taken forward. Out of these 19 embryos, 2 were implanted in the mother to give rise to the twins. These concerns over Lulu and Nana's well-being are only one of a myriad of issues. Perhaps most critically, there was no medical need for this treatment in the first place. It has been claimed that the biological father of the twins is HIV positive, but given that the couple used IVF and that there is no evidence that HIV would be passed on from father to child using this method of conception, the procedure was medically unnecessary. Furthermore, He claims to have induced this mutation completely into at least one of the twins. To do this, He and his group would have had to check the DNA of every cell in the growing blastocyst, a group of cells that exist from days 5 to 9 post-fertilization that eventually becomes the embryo. Given that they would have had to destroy the blastocyst to test all the cells, a clear logistical problem exists in trying to qualify the mutation exists in all cells. Accordingly, He and his group only checked a number of cells from the blastocyst. This means both twins might have a condition called daism, where the deletion only occurs in a subset of their cells. This means that the different cells in their body have different DNA. In this context, the effects of this condition are completely unstudied. Finally, the gene that He took aim at has also been reported to have a role in learning. A link between the activity of the CCR5 gene product and the dampening down of the brain's ability to form new neural connections has been previously described in mice models. Theoretically, there is a possibility that the induced deletion could have a positive effect on the twins' ability to learn. Right now, there are far too many unknowns to make an accurate prediction on what, if any, effect this experiment will have on the two twins. And there is an even bigger question about what effect the announcement will have on the progress of the field. Complicating this mess is the fact that this study did not go in front of an adequate ethics approval board, and the default Chinese laws prohibits any kind of follow-up with the twins. Because of this, we will likely never know what will become of Lulu and Nana. Dr. He has stated that he plans to publish the work in a peer-reviewed journal, but given the fact that he has gone MIA for almost 2 months now, the scientific community may never have an opportunity to properly review and digest the impact of his work. So where do we go from here? There is no doubt that genetic engineering is a valuable tool that can potentially safely increase human well-being and productivity. It's not unlikely that plunging into these uncharted waters unaccounted and without approval may have a chilling effect on proper utilization and development of this technology. Perhaps until a consensus on the way forward is established, this is a good thing. Thankfully, the World Health Organization has stepped up to take a moderating role in the process and are establishing a working group to establish clear guidelines. We need these guidelines because we've stepped into a new era of medicine, one where the genetic modification of humans is not only a possibility, but an